Bismillah ar-Rahman ar-Rahim. The title of our lecture is a complement system. We'll deal with the structure of the complement system, the activation, and the function. The complement system, the abbreviation of the complement system is a capital C. The complement components are proteins or glycoproteins that are synthesized mainly in the liver, hepatocytes, monocytes, macrophage, and epithelial cells. So this is the source of the complement. The function of the complement, as its name, is to complete the action of the antibodies to eliminate the pathogen. So it's fix the complement and to complete the action of the fix the antibody and complete the action of the antibodies to eliminate the pathogen. It's present in the blood serum. It's designated by the numerals C1, C2 to C9, numbered in the order of discovery, not sequence of action. And also it had symbols, letters like capital D, capital D. About 15% of the globulin fraction. The complement constitute about 15% of the globulin fraction. The complement destroyed by heating because it's a protein, so it is denatured by the exposure to heat, and the serum does the complement when it's heated at 56 centigrade for 30 minutes. Its complement circulate in the serum in the inactive state. Once it is activated, a peptide fragment will be formed and donated by small letters. A small fragment designated A and large fragment designated small b. Complement activation. It's occurred through the classical pathway, alternative pathway, and the lectin pathway. First, we have the classical pathway. It's part of the adaptive immunity. It's activated through the binding of the antibody, which is the IgM, IgG3, IgG1, IgG2, plus antigen, which is called the immune complex. Immune complex is antigen plus antibody. And the Fc portion of the immunoglobulin recognized by the C1. C1q binds to antigen-antibody complex, then C1R, then C1S, which is lead to the C1QRS. So this is the classical pathway. This is the antigen and antibody, and this is called the immune complex, which is initiate the classical pathway. So this antigen-antibody immune complex will bind to the C1q. C1q, which is C1 is consist from C1QRS. So this is lead to binding of the antigen-antibody to C1q. C1, it is a macromolecule complex present in the serum consisting of three subunits C1q, C1R, C1S, connected to each other by the calcium dependent bonds. So this is like the tulip flower. It consists from the Q, R, and S, and it's bound to the through the calcium dependent bonds. Antigen-antibody activates C1q, then C1R, then C1S. C1S has enzymatic activity, which is esterase activity, cleaves C4 into two fragments, C4A and C4B. So this is the antigen-antibody, which is the IgG, activates C1, and then C1 cleaved to C1QRS, which is cleaved C4, which is C4A and C4B. C1S in the presence of C4B will split also C2 into two fragments, C2A and C2B. This is C4A, C4 is cleaved, and C2 also cleaved into C2A and C2B formation. Lead to formation of C3 convertase, which is C4B2A, that cleave the C3 into C3A and C3B, forming a complex called the C5 convertase, which is C4B to A3B. C5 is cleaved by C5 convertase, which had enzymatic activity that cleaves C2 into C5A and C5B. C5B attached to cell membrane to form C4B to A3B5B act as a receptor to C6 and then C7, then C8, then C9. So this is the C5 divided into C5A and C5B, and this need to bind to C6, C7, C8, C9, bind to the cell membrane, form complex which is called a MAC, membrane attached complex, causing pore on the cell surface, and this leads to influx of sodium and water and lysis of the cell. And this is lead to formation of the pore in the cell wall and lead to influx of the extracellular fluid, which lead to lysis of the and rupture of the target cell. The other fragment, smaller fragment, the function of C3A, C3A and C4A, which is act as mediators of anaphylaxis and chemotactic substance to other immune cells. C3B, which is immune adherence and function in opsonization. It's important in the opsonization and enhance the opsonization. This is the classical pathway of the complement activation. The second pathway, which is the Mannose Binding Lectin pathway, which is the M for the mannose, B for the binding, and L for the lectin. Mannose Binding Lectin is a serum protein that binds to mannose in the microbial cell wall. So this is Mannose Binding Lectin, which is the serum protein present in the serum, bind to mannose, which is present on the cell wall of the pathogen or the bacteria. It's part of the innate immunity. This complex has function of the C1QRS, and this complex cleave C4 and C2 and continue to C9. So the difference from the classical is that instead of C1QRS is the Mannose Binding Lectin. The another pathway, which is the alternative pathway, which can be initiated by three distinct ways, which is takeover pathway, and it can be a takeover pathway, which is spontaneous hydrolysis